Okay, so hello and welcome everyone to today's live class session of week 8. And today I will be mostly discussing on metalloenzymes and metalloproteins and different motor proteins, and then discussing with you a few question and answers which will help you in solving the assignment and taking up the future exams. So I am Srijan Sen from IIT Kharagpur, and this course is being taken by Professor Shagota Dasgupta of IIT Kharagpur. So let us start with the lecture. I am sharing my slides with you all. Okay. So, in the previous class, we discussed, in the previous two classes basically, we discussed about the enzymes, their classification, and their kinetics. We discussed about how the ligands are binding to the enzymes and different active and the catalytic sites. And what are the different types of sites which are present in the enzymes? What are the different classes of enzymes that exist in nature, and how do they function? What are their functions? What do they play a role? And then in the next class we spoke about the kinetics, the enzyme kinetics, and different inhibitors or the different types of inhibition kinetics, that we spoke about all of these particular things in the previous classes. So in today's class, we would be mostly focusing on the metalloproteins and the motor proteins. So when we speak of metalloproteins, what basically metalloproteins actually are, that is very important to know. So what are these metalloproteins? So from the term, if we can understand that there is a kind of a metal that is actually playing a role in the proteins. So what can we tell? So metalloproteins is a generic term for a protein that contains a metal ion as a cofactor. So previously we have heard about apoenzyme, then holoenzymes, and how the cofactors or different activators are actually activating the enzyme to play a particular role. So these metals are actually acting as your cofactors. So a large proportion of proteins basically falls in this category. So I would be mostly focusing on a few of them, and then when I would be discussing with you the questions, I will be coming across a few more examples of that. So some examples of these metalloproteins or metalloenzymes includes your urease. So that is one of the example. So one example is urease, that actually catalyzes the hydrolysis of your urea into carbon dioxide and ammonia. So the reaction occurs as this, it is given over here. So now you can see over here there is another enzyme, that is aconitase. So what does this what is this aconitase? So it is basically an enzyme that catalyzes the stereospecific isomerization of citrate to isocitrate via your cis aconitate in the tricarboxylic acid cycle, or which is commonly known as the TCA cycle. And this is mostly, you can call it as the non-redox active process. So basically, if we tell about what is the function of an aconitase enzyme, so it is basically it controls the metabolic flow and the ROS production. So these mitochondrial aconites can be oxidatively inactivated by loss of some labile iron or by the oxidation of the cysteine residue. So it needs a particular ion. Okay, so what is the ion, or what is the particular cofactor that is playing a role over there? So you need a particular say a cluster. So that cluster is of Fe4, that is iron 4, and sulfur 4, which has a charge of +2. So that is the active form that activates this enzyme to perform its different functions. So here you can see that there are a lot of enzymes that are playing a role over here. So these are the known biochemical functions of selectable trace elements that are known to account for the sizable fraction of cellular metals in the marine organisms. So from my theory portion, I would be discussing a few examples, and when I will be discussing with you the question and answers, or solving the problems, then I will be telling you a few more examples, then you will get to know. Okay, so here we have the metal manganese that's function as oxygen evolving complex of your photosystem 2, or the super oxide dismutase. Then we have iron, which has different Fe ferrous sulfate centers, for example your aconites, then ferredoxins. Then you have your cytochromes, that's very common. Then you have the nitrate reductase, the nitrate then the superoxide dismutase. Then you have cobalt for carbonic anhydrase. Then for urease you need nickel. For you need copper for plastocyanin, ferrous oxidase, amine oxidase. You have zinc for carbonic anhydrase, and cadmium for carbonic anhydrase, and alkaline phosphatase. So these are the common metals that act as cofactors to carry out different functions in your different enzymes, or you can call different proteins. So what is the role of metal in biology? So if you see that metalloprotein functions in your dioxygen transport, basically these proteins falls in the category of your hemoglobin, myoglobin family, then your hemocyanins, hemerythrins. Then it plays different roles in the electron transfer, like your nitrogen fixation. Then it has different structural roles in your zinc finger motifs and all. Then what is the role of the different metalloenzyme functions? So these metalloenzymes, the metalloproteins that perform a catalytic function. So like your hydrolytic enzymes, like your carbonic anhydrase. Then you have the nitrate reductase, oxidation of hydrocarbons by the photosystem P450, or the two electron redox enzymes. Then you have the multi-electron pair redox enzymes, like your cytochrome C, photosystem 2 nitrogenase. And finally, this plays different roles in the rearrangement mechanisms, like your vitamin B12. So there are different roles of metals in biology. So few of them are like they have different roles in the photosynthesis. So this is basically the diagram of your photosynthetic apparatus of oxygen phototrophs, which is speaking about the proteins that contain iron with the potential of electrons at different steps, which are present over here. Now also what are so metal enzymes in photosynthesis. So iron content of proteins or protein complex involved in the photosynthetic electron transport. So you see the protein or the protein complex over here, and the number of iron atoms per protein complex. So if you see photosystem 2, it has around 2 to 3. Then photosystem 1 has 12. So cytochrome B6 has 5, B6F has 5, cytochrome C6 has 1, then you have ferredoxin, which needs 2, catalase 4, then iron superoxide dismutase 1, ascorbate peroxidase 1, NADPH, PQ, plastoquinone, oxireductase needs 8 to 12, 8 to 18. So these are the number of iron atoms that needs to actually increase the catalytic activity of your proteins, or that act as the cofactors for your different proteins or the protein complexes. So this is another example where metal enzymes play the different roles in carbon fixation, and where they play a role. So you see there is role of zinc, cadmium and cobalt, whereas here you need silicon, followed by here you need rubisco. So these are the different things you had studied when you had studied these particular mechanisms. Now metal enzymes also play different roles in nitrogen utilization. So for that you need the first one being the nickel, then you have different you need the iron, molybdenum, then you need another iron over here, and in different steps, or in different to perform to catalyze different steps, you need different metals, and these actually catalyze your the activity of different kinds of say mechanisms which is occurring in nature, or the different steps which is occurring in the nature. Okay, so basically you won't be getting questions from here. These are just a few examples when I will be coming to the assignment, because today's assignment is very important, because what is not there in the theory will be there in your assignment, or in the questions that I will be discussing. So in there also I will be discussing as to what type of questions you can expect. Okay. As of now, let's focus on the few examples that occur in nature, and the different kinds of metals that play in the different naturally occurring reactions. Now, metal enzymes in the nitrogen biogeochemical cycle. So if you see over here, you need in the nitrogen fixation, you need iron and molybdenum. Then in the ammonium oxidation, you need iron and copper. Then in nitrification, you need iron. For denitrification, you need the role of iron, copper, then iron, and finally copper. So these are the different. So this is the diagram of the nitrogen cycle with the different catalyzing enzymes and the metal requirements of each step. Okay. So and the abbreviations of the different short terms are given over here. You can get it from my slides, so I will be not much focusing on this. Let's jump into motor proteins. So when I will be discussing the assignment, I will be giving a few more examples as well. So when we speak of motor proteins, so there are actin based motility, where motor proteins are basically your myosins. Then you have the tubulin based motility, or motor proteins, that are your kinesins, then dyneins. And the roles of the actin based systems, basically you have the cell crawling by the lymphocytes, you have growth cone extension, then muscle like contraction of ovarian follicles, then mammary gland ducts, etc. Then you have different muscle contractions, distribution of vesicles. So these are the different roles of the actin based systems over here. So and then you have the intermediate filaments and organelles within the cytoplasm. So these are the different actin based system, the roles of different actin based systems. Now the actin based motility, the myosin, so the head is both actin binding and the ATP binding, where the purple chain has a regulatory role, so it regulates different kinds of functions which these myosins actually perform. And the myosin 2 is a muscle myosin that actually plays role in your muscle contraction of the ovarian follicles, the mammary gland ducts, and a different muscle contraction for growth of different muscles. So if you see over here, this is the actin binding domain. Here you have the light chains. This is the head portion. You have the neck portion where you have the regulatory junction, and then this is the tail portion. So in the tail portion of myosin 2, you have two heavy chain tails coiled around each other, and for myosin 5, you see you have two heads. Although you have two heads and two neck over here, and the tail portion is coiled. Whereas for myosin 5, the tail portion is coiled, and again they open up. So this is the different kinds of myosins present. So the study of the movement of myosin against your actin filaments and in vitro motility assay. So preparation of actin cables pointing in the same direction. So these are the used cells from a giant alga that uses these cables for moving chloroplasts around. Then you have the open algal cell, and you add yellow fluorescent beads which is coated with myosin, and then you add the ATP and take time-lapse photography. So by this method actually you can study the movement of myosin against your active filaments via the in vitro motility assay. So this is a much of an experimental method as to how you can do. So in the first step, what you do, I'm just repeating it once more, is you have to prepare the actin cables which are in the same direction, because as you see from the structure they have to be in the same direction. Okay, and these are the use cells from the giant alga or any organism that you take that uses these cables for moving the chloroplast around, and then you add the yellow fluorescent beads coated with myosin, and then add ATP and take photography or click pictures at different point of time. So this is the cartoon structure of the myosin coated bead actin preparation, where you have the bead, you have the S1 head, and here you have the this is your myosin, this is your head, and this is the actin. So here, from here you can understand how the myosin is bound to the bead and finally to the actin. So how do you study the speed of mode or speed of movement? So myosin coated beads move unidirectionally, and it's mostly an ATP dependent process, so it requires some kind of energy. So the energy is obtained from the ATP molecules, and the speed of the beads coated with the muscle myosin is mostly your 5 micron per second, which is the same as that of the contraction of the sarcomeres in the muscle. So they have to be in so the speeds have to match one another to be able to perform its function. So the speed are more or less similar to that of the, means the beads coated with muscle myosin is similar to that of the contraction of the sarcomeres in the muscles. And different myosins are definitely, there are exceptions, and different myosins move at different speeds. So the smooth muscle myosin moves at 1 micron per second. So the speed of movement of the myosins determines where they would be present or where they are located, because some the myosins cannot move at a very high speed even if they want to, or they cannot move at a very low speed even if they want. So the speed plays a very important role for the movement of the myosins, and these are the of the common muscle myosins which are present in our body. So the studies of movement due to a single myosin molecule, so how do we do that? So we use a setup in which the focused laser beam creates optical traps. So you see there is a laser beam, you have a bead over here, and then you have the myosin which is connected, and you have the glass slide. So these optical traps can hold these small objects. The force is controlled by adjusting the intensity of the laser beam over here, and you pass the laser beam through the bead, and it goes and strikes the glass slide. So what you have over here, so the actin filament is held in the optical trap via 1 or 2 attached beads. So you see you have the myosin over here, and these are the actins and all. So the laser beam is passing, and it's striking the glass slide. So the myosin concentrations are kept low so that only one myosin contacts the actin filament. So because of to ease for the ease of study, that can be done. Okay. Now also, so the first thing that we need to do is we have to use a setup in which the focused laser beams create an optical trap. So that optical trap has to be created so such that it can hold the small objects, and then you have to, the second thing is that you have to modulate the force such that the intensity, you have to change the intensity by adjusting the force. The second thing is that the actin filaments is held in the optical trap by 1 or more attached beads. So that is okay. The third thing is that the myosin concentrations have to be low such that only one myosin contact with the actin filaments. The next thing is that the ATP has to be kept low such that only one ATP binds with the one myosin head. So one myosin concentration is kept low such that one myosin binds with one actin, and the ATP is kept low such that one ATP binds one myosin bead. And in this way you can get the result. So if you see that in the y-axis you have the displacement, in the x-axis you have time. So at different time you click the pictures and you understand the displacement. So you would see that the results show that the myosin pulls on the actin filament in a stepwise or a ratchet like fashion, and in this way you are going to get the graph. So a single ATP molecule is hydrolyzed, resulting in a power stroke and displacement of around 10 nanometer. So you see that here you plot the distance in nanometer, and in the x-axis you have time. So for, let's say, for 1 second you take the sample, and here you have different bursts or peaks, and this is your power stroke, because after you get a sudden strike. And then over here there is a release, because the distance is reducing with time. So this is the release point, and this is your power stroke point. And then if you zoom into the picture, or plot force versus time, then you would see that the force generated can also be determined, and it ranges around 3 to 7 piconewtons. So in this way you can understand what are the different factors that are important for your understanding the movement of your actins and the myosins. So the force is generally between 3 to 7 piconewtons, and here you plot force in piconewtons versus time in seconds. Okay. So this is the distribution of a vesicle along the actin network that's polarized. So you have the pointed ends over here. This is your actin filament, and these are your barbed ends. So this is your myosin 1 head. This is your tail portion, and this is the membrane vesicle present. Now if we put it together, so the actin and the intermediate filament, then what happens? Let's take the example of myosin 5. So the myosin 5 has two head portions, and it has a coiled intermediate region, and then another point over here. So what we have, this is the head portion, this is the pointed end, and this is the barbed end. So your myosin 5 is the link between this actin, which serves as the rigid skeletal element, and the intermediate filament, which is being delivered to the another part of the cell. So here you have the head of myosin 5, which is attached to the one to the pointed end and one to the barbed end, and then you have the coiled portion, followed by the light chain, which is present over here, which is attached to the intermediate filament fragment. So that is all about your actin and the intermediate filaments. Now let us talk about tubulin based motility. So what is the different role of the tubulin based system? So one is the exoplasmic transport, then you have in the growth of the mitotic spindles, then they have different roles in the growth of cilia and in the movement of the flagella in different kinds of cells or unicellular organisms. Then there are positioning organelles within the cell. So this, just because they are motor proteins, they help in the movement basically. So different places where the organelles needs to be positioned, so they function over there. Also they help in the movement of the entire cell via the growth of your mitotic spindles, and by the regulation of the cilia and the flagellar movements, and also in the exoplasmic transport. So what is exoplasmic transport? So we have the anterograde from cell body towards the synaptic end, that is towards the positive end of the tubule, which is driven by your kinesins. Then you have the retrograde from synapse towards the cell body, which is driven by the dyneins. And how fast do they function? So the fast they work for the cargo carried in vesicles is 50 to 400 millimeter per day, and slow for individual protein molecules, that is net rate is less than 10 millimeter per day, but apparently this reflects a stop and goes aspect of the process. So it can be a fast depending on what type of work it needs to be carried out in the vesicles, and that can be up to 50 to 400 millimeter per day. And also if the net rate can be as less than 10 millimeter per day if we look into for the individual protein molecules. So speaking of the functions of the exoplasmic transport, so it is basically important in delivering your proteins, different organelles like your mitochondria, vesicles to the synaptic positive end of the synapses. and it removes the recycled proteins or organelles to the cell body for destruction by the lysosomes, because these dead cells or dead organelles are no needed, so they need to be recycled. So that is done via this exoplasmic transport, and then it carries out different intracellular chemical messages from the synapse to the cell body, like different signals and feedback messages and all. And finally, it delivers the neuron specific viruses like your herpes and the pox virus from the peripheral sensory nerve endings to the cell bodies in the central nervous system. So here you see that microtubules are associated with the motor proteins like the dyneins and the kinesins. So kinesins are thought to have evolved from the myosin. So this is, you have the pointed end and the barbed end, and then this is the head domains of the dyneins and the kinins. And this is the coiled coil. Here you have the light and the intermediate chains and the light chains. So the head portion actually joins with the different ends. Okay. Okay, so this much is all about the theory. Now when I will be now let's discuss with the questions. So when I will be discussing, you can write down the different points that I would be telling. Okay, so today's assignment I would be only discussing, or would you like to give the answer? What's your suggestion? Would you like to answer, or you want me to discuss? Because we have only one student, so we can wrap up early. You want to answer? Okay, let's see. If you want to try, let's see. So which of the following is or are true? So myosins are actin-based motor systems, dyneins are actin-based motor systems, myosins are tubulin-based motor systems, and kinesins are tubulin-based motor systems. So what is what? D, you said 2 and 3. So it is saying which of the following is or are true. So let's see. Okay. So for the first one is myosins are actin based motor systems. So that is correct. So myosins are actin based motor systems. The next one is dyneins are actin based motor systems. That is not correct, because dyneins are tubulin based motor systems. Okay, then kinesins are tubulin based motor systems. That is also correct. So, and you have the myosins that are actin based, so myosins are not tubulin based. So 2 and 3 are incorrect. So actually 1 and 4 are the correct options. So C is correct. So myosins are actin based motor systems, dyneins are tubulin based motor systems, and kinesins are tubulin based motor systems. Okay, the next one. Which of the following is or are true? Dyneins show retrograde transport. Kinesins show retrograde transport. Kinesins help to move the chromosome along the microtubule, and the ATP bound state of kinesin weakly binds with the microtubule. So which do you think is or are correct? Okay, you say B. Okay. So let us see what are the options. So the first one, it is saying dyneins show retrograde transport. Yes, that is correct because dyneins show retrograde or the negative end transport. So one is correct. So let us see in where we have one. So it can be 1 3 or 1 4. Okay. So kinesins show retrograde transport. That is not correct. That is incorrect. Kinesins help to move the chromosome along the microtubule. That is also correct. And the ATP bound state of kinesin weakly binds with the microtubule. That is not correct. They strongly bind with the microtubule. Therefore 1 and 3 are correct. A is the correct option, not B. Because ATP bound, we have to understand it that it is not weakly binds. It is incorrect. It strongly binds with the microtubule. Okay. Now, can you identify X, Y, and Z? So, what is X, what is Y, and what is Z? Let us see if you can identify them. And also, you have to identify the image. So, you have to identify basically 4 things. One is the image, one is X, one is Y, and one is Z. Let us see. Quite tricky options. So these type of questions you can expect where you would be getting a few figures and all. Okay, so I'm just telling you the answer because here is nothing to basically explain. So let us first see what is X, Y, and Z. From there only we can understand what is the image. So X, it is radial spokes. We all in the our options are radial spokes. So radial spokes is the correct option. Now the next one is doublet microtubule and the quartet microtubule. So let us see what is present over Y. So what do we have over there? So Y is basically your doublet microtubule. So it is quartet. So C is wrong because the X is radial spokes, Y is doublet microtubule. The last one is Z. So okay, so Z is pointing somewhere else. Okay, I think basically it is pointing to the this portion. So Z is basically pointing to this portion. So this is some arm, and this arm is your dynein arms. So radial spokes, doublet microtubule, and dynein arms. So this one is, do we have somewhere else dynein arms? Okay, so just because it has the dynein arms, so it is going to be axonymal dynein. So X is your radial spokes, okay, Y is doublet microtubule, and Z, that is your arm, is your dynein arm. So that is your axonymal dynein. Maybe you will not get this kind of questions because it's difficult to understand even for me from this particular picture. But as of now, D is the correct option. So see, you have the, because of the dynein arms, it is the axonymal dynein. If the answer would have been kinesin arms, it would have been axonymal kinesins. So D is the correct option. Okay. Now tell me, which of the following is true? If you know the answer, it's okay. If not, I will be discussing because because today's assignment is more of a theory class only, because these are the questions which I have not discussed in the theory, and it's good to discuss from the question and answers. So tell me, myosins are in. Which of the options are true? So myosins are in a high energy state when the head is bent at 90 degrees. Myosins are in high energy state when they are bent in 45 degrees. Actins are in low energy state when they are in 45 degrees, and myosins are in a low energy state when they are in 90 degrees. So which of the following option is true? So you say actins are in a low energy state when the head is bent at 90 degrees. So does it actually depend on actins? Basically no. It is totally on the myosins, whether they would be in the high energy state or the, because see, actins are a linear kind of a thing, so in what way they are bent, it is not much of an issue. But when I was speaking of the myosins, what I was telling, that if they are in a high energy state, when they actually, saw that for myosin 5, okay, so they were, one was attached to the pointed end, one was attached to the barbed end. Okay, so the angle which they were doing was actually 90 degrees. So your myosins are in high energy state when the head is bent at 90 degrees. But but all of the others are wrong, because no information about actin is known, and myosins are in low energy state when they are bent at 45 degrees. So this is wrong. So from the myosin perspective, if we tell that they are in a high energy state when the angle between the two heads is actually 90 degrees, just as we observed for myosin 5. Okay, never mind. The next one, which of the following is false? ATP synthesis takes place when stroma has high proton concentration relative to thylakoid lumen. Gamma acts as an endogenous inhibitor for ATP synthase. ATP synthesis takes place when gamma shaft rotates clockwise, and ATP synthesis takes place at beta subunit. So which one option is wrong, or which other three options are correct? Because this is a question on ATP synthesis, and there might be a question on ATP synthesis even in your exam, or might be in your assignment, I do not know. Okay, very good. So the first option that it says, ATP synthesis takes place when stroma has high proton concentration relative to the thylakoid lumen. That is a correct option. Okay, so this option is correct. So ATP synthesis takes place when the gamma shaft rotates clockwise. That is also correct. So we had studied about that. ATP synthesis takes place at beta subunit. This option is also correct. But what is not correct is gamma acts as an endogenous inhibitor of ATP synthase. This is the option that is incorrect. So what is the correct answer to this? It is B, but what would be the answer? It is eta. Eta acts as an endogenous inhibitor of F1 by blocking your gamma rotation due to the steric hindrance. So eta is the main factor over there, or it is the endogenous inhibitor of your ATP synthase. So how is it doing? It is blocking the gamma rotation due to some kind of a bulkiness or the steric hindrance. So B is the correct option, means the correct option for this, because it has asked me to tell the wrong one. Okay, this is one question from your metalloproteins, or you can tell metalloenzymes. So the image shown above is an active site of cupridoxine. Okay, so the geometry of this is, so what is the geometry of cupridoxine? From the figure, what can you tell me from about the geometry? So from the figure, what can you understand? So here you can understand that cupridoxine is tetra coordinated with copper. Okay, so cupridoxine is only the entire tetra coordinated copper in its active site. So it is tetra coordinated. Okay, and then you have the two ligands that are in histidines, means the N of the histidine is bound, and one sulfur from the cysteine at the base, with one sulfur of methionine in the long axis. So you have one in the long axis from methionine, you have one in the base with cysteine, the sulfur of the cysteine, and then of the two histidines. No answer? So, sorry. So actually the geometry is trigonal pyramidal. So trigonal pyramidal is basically like this. One ligand over here and one ligand over, and then you have one here, one here. So you have 5 ligands, but here you can have something called maybe lone pair, or it can be empty, but this is your trigonal pyramidal structure. Okay, so one, you have in this position you have the cysteine, sulfur of the cysteine. In this position you have your methionine, which is also with the sulfur, and in the other two positions you have the nitrogens of the histidine. So it is giving you a trigonal pyramidal structure. The next one, which of the following is false regarding hemocyanin? So you people had studied about hemocyanin. So which is the statement which is false? It has two copper ions in its active site. Copper ions are in +1 oxidation state in the deoxy form. Each copper is coordinated by two N nitrogen atom of the histidine residues, and the deoxy form of hemocyanin is colorless. So one is the wrong answer. Others are correct. Tell me which one is wrong. Okay, let us see. So it has two copper ions in the active site. That is correct. The last one is the deoxy form of hemocyanin is colorless. That is also known to us. That is also correct. So the copper ions are in +1 oxidation state in the deoxy form, and the other one is the copper ion is coordinated with two nitrogen of the histidine residues. So which one do you think is incorrect? This one is incorrect because it is not 2, it is 3. So 3 nitrogen nitrogen atom of the histidine residues are involved over here. This is correct because the copper ions are in the +1 oxidation state in the deoxy form. But this is the wrong answer. So for you the correct answer would be C, because that is the wrong answer. Because 3 nitrogen atoms of the histidine residue are actually involved. Okay. Now another one. Which statement is false regarding myoglobin and hemoglobin? Oxygen saturation is higher and at low pH for myoglobin. 2,3 BPG increases oxygen affinity for hemoglobin. Iron binding site are similar for both myoglobin and hemoglobin, and oxygen saturation is higher at low partial pressure for oxygen for myoglobin. So which one is incorrect? In I think in the last class, or maybe 2 to 3 class before, we spoke about myoglobin and hemoglobin in details. So if you remember, you can answer this question. And 2,3-BPG is a ligand that basically helps in better oxygen transfer from your hemoglobin to myoglobin. Okay, so now tell me, I have already given the hint. 2,3-BPG is actually so what is the correct answer? Means which of the following options are incorrect? So let us see from the first option. Oxygen saturation is higher at low pH for myoglobin. So if we see that plot, we can tell that this is the correct option. Oxygen saturation is higher at low partial pressure of oxygen for myoglobin. That is also correct. Now the other two is oxygen binding sites are similar for both myoglobin and hemoglobin. This is an obvious one, and this is also the correct option. What is incorrect is this. 2,3-BPG increases oxygen affinity for hemoglobin. Why is it incorrect? Because it does not increase, rather it decreases the oxygen affinity of hemoglobin. Thereby what it does, it helps in the oxygen transfer from the hemoglobin to myoglobin. Therefore this is the incorrect option. It decreases the oxygen affinity. Now tell me the enzyme and its cofactor involved in this conversion. Okay, so we need to find out the name of the enzyme and the cofactor that is involved in this particular conversion or this reaction. So first we have to understand what is the name of the enzyme that is performing this reaction. So basically it is adding a phosphate group. So which enzyme adds a phosphate group? No, that is not correct actually. So phosphate group is added by kinases. For phosphatase, it is an ase that is breaking. Phosphatase breaks the phosphate group. Kinase adds a phosphate group. So kinase is the right answer. So we have hexokinase over here. So it has to be either magnesium and or the porphyrin ring. So, what is the role of hexokinase? So, hexokinase, if you see, converts or converting glucose to glucose 6 phosphate. So, this is your glucose, and this is your glucose 6 phosphate. So, it is converting in the presence of your magnesium ion. So magnesium ion is playing the role of a cofactor over here. So the correct answer is A. So remember that kinases adds a phosphate group. Hexokinase means in the sixth position. Glucose phosphate to glucose 6-phosphate, and phosphatase breaks the kinase, breaks the phosphate group. Okay. So A is the correct option over here. Okay. Now let's come to match the following. Let us see what do we have over here. So we have catalase, peroxidase, rubridoxins and the ferridoxins. So which one is correct? So we have to find the function. Yes, this is easy if you know the for match the following, the easier thing is that if you know at least two of the correct answers, that will actually help you to find out the entire thing, all of them. Okay, let us see what is your answer. Okay, you are telling 4 is B. Okay, that is correct. That is correct. So it can be either A or D. So tell me which one do you think is right. So 4 is B means ferredoxins. Ferredoxins have multiple iron centers. So let me, oh sorry, let me mark over there. So yes, so ferredoxins B. Yes, that is correct. So then you have 3, that is D. Yes, 3D. So rubridoxins does not have any inorganic sulfur. That is also correct. Okay. Now, what about 1 and 2? You have the catalase and you have the peroxidase. So what does peroxidase do? So peroxidase catalyzes oxidation of wide varieties of organic and inorganic substrates by H2O2. So what it does, it catalyzes the oxidation of different organic and inorganic substrates by H2O2. And catalase, on the other hand, catalyzes the disproportionation of H2O2. Okay, so what is the correct option? The correct option is D. So 1A and 2C. So 1 is catalyzing the disproportionation of H2O2 and 2 is oxidation of organic substrates by H2O2. Okay. Okay, never mind. We have, okay, another one. Let us see. Which of the following is or are false regarding azurine? It has 2 copper centers. Then it undergoes oxidation reduction between copper 1 and copper 2. Each copper is coordinated by 2 nitrogen atoms of histidine and 2 cysteine residues, and the oxidized form of azurine receives only 1 electron. Which of them is incorrect? It's okay if you do not know, because I have also not thought about azurines. So azurines is a tetrameric protein, okay, and it has 4 copper centers. So those copper ions are coordinated with 2 in histidines and 3 cysteine residues at the equatorial position, and the carbonyl oxygen of the glycine and the methionine. Okay, so first of all, it's a tetrameric protein and it has 4 copper ions. So 1 is incorrect because it is saying 2, so it is the incorrect option, the first one. Then it undergoes oxidation reduction between 1 and 2, that is correct. Now each copper is coordinated by 2 nitrogen and 2 cysteine residues, that is incorrect because it is coordinated by 2 nitrogen atoms of the histidine and 1 cysteine residue and the carboxylic carbon of the glycine and the methionine, and the oxidation form of azurine receives only 1 electron. This is correct. So 1 and 3 are incorrect. So this is the right option for this question. The next one. Okay, what it says, the plot of saturation fraction and partial pressure of oxygen of hemoglobin at different pH 1, 2 and 3 is shown. The order of 1, 2 and 3 will be, okay, so what is the order of 1, 2 and 3? So this is theta, that is fraction saturation, saturation fraction, sorry, and your partial pressure of oxygen of hemoglobin at different pH. So as far as I can understand from the plot, low affinity is for 3 and high affinity for 1. So can you tell me which is the correct option now? Very good. That is the correct option. Because at lower pH, oxygen affinity of hemoglobin decreases, and number 3 is showing the lowest pH plot, because it is at the lowest oxygen saturation at at that particular pH. So for 1 it is the greatest, and for 3 it is the lowest. That is 1 greater than 2 greater than 3. Very good. Okay. Now these are two clusters, pictures of two clusters that is given. So find out the correct statement. Electron transfer takes place from cluster 1 to 2 and N2 binds to cluster 1. Electron transfer takes place from cluster 2 to 1 and N2 binds to cluster 1. Electron transfer from 1 to 2 and N2 binds to cluster 2, and from 2 to 1 and N2 binds to cluster 2. Okay. Similar type of options. So two things you have to find: where will that N2 go and bind, and from where will the electron transfer take place. So you are saying number A. So the electron transfer will take place from the FEMO motif of cluster 1 to cluster 2. So that is correct, and N2 will bind to cluster 1. Yes, that is the correct option. So electron transfer takes place from this motif to this motif, and N2 will go and bind with the cluster 1 and get reduced to your NH3. Yes, that is correct. So we have the last one for the day. That's also match the following. So find the function of polymerase, helicase, gyrase, and ribosome. It's an easy one. Very easy, I think. Reads and translate mRNA to polypeptide, catalyze DNA supercoiling relaxation, separates two annealed nucleic acid strands, and catalyzes phosphodiester bond formation between nucleotides. Okay. So this is easy. You can tell me the answer. So what is the role of ribosome? If you know what is the role of ribosome, I think that will be Easy for you to answer. What does ribosome do? And also, what does polymerase do? Polymerase activity should be known to all of us. We have spoken a lot about polymerase. We have studied DNA replication, transcription, translation. From there, the question is very simple. So if we talk about the activity of polymerase or the role of polymerase, so there are two types of polymerases. One is the DNA polymerase and the other one is your RNA polymerase. Okay, so the DNA polymerase catalyzes your addition of nucleotides onto the existing strands, and your RNA polymerase uses the single-stranded DNA template to synthesize a complementary strand of your RNA. So it catalyzes the phosphodiester bond formation between the nucleotides. So one is D. So we have only one option for that, so that is B is the correct option. Let us see what does helicase do. So helicase utilizes the energy that is derived from ATP hydrolysis to move along the nucleic acid and to separate two annealed nucleic acid strands. So what is that option? Yes, so separates two annealed nucleic acid strands. So that is your C. So 2C. So 1D, 2C. The next one is guidase. So DNA guidase catalyzes in DNA supercoiling or relaxation, and it introduces negative supercoils into the DNA at the expense of your ATP hydrolysis. So it catalyzes your DNA supercoiling as well as relaxation. And lastly, your ribosome, that we should all be knowing, it helps in the translation of your mRNA to the polypeptide. It reads the mRNA sequence and it helps in the translation of proteins. So 1D, 2C, 3B, and 4A. So that was all about this. Thank you. Do you have any questions? If not, we can just end the class. Okay. Thank you then. We will again meet in the next week with a different lecture. Okay. Thank you all.